Here you can see a section of a high resolution electron microscope that is taken from the testes of a mouse. These greenish dots that you can see in this micrograph are spermatogonia. And you know that in spermatogenesis process, of course, I'm not going to revise it, but in the spermatogenesis process, these spermatogonia, they will enter into meiosis, right? These spermatogonia, first of all, they will divide, right, and they will produce two type of cells. The some cells will stay near the basal lamina, while the other will go into the deep lumen, and they will enter into meiosis in the reduction division, right? They will complete meiosis 1, that is the reduction division. They will complete then the meiosis 2. They will then enter and they will form into spermatids. These spermatids will differentiate into mature sperms, spermatozoa, right? This micrograph is all right, it's very good, but the problem in this here in this micrograph is these small dots, these reddish dots. Now these reddish dots are cytoplasmic bridges that are connecting the cytoplasm of these two spermatogonia with one another. But the problem here is that why do we need these cytoplasmic bridges in the developing process or in these steps of spermatogenesis? Because when I see oogenesis, right, I don't see the oocyte that is connected with one another with some similar junctions that are connecting the cytoplasm of the two oocytes with one another. Why do we need cytoplasmic bridges for spermatogenesis? And why do we not need it in, oogenesis? What is, what are their significance? Because if they are present, so they are present for some reason, right? So these cytoplasmic bridges, in order to understand this, I think first of all we should know the differentiating system that is in humans, because of course now we are talking about humans. We are not talking about mouse anymore. So in humans, males are heterogametic, right? Males are heterogametic. Let me use a different color. Yeah, males are heterogametic, meaning to say the 23rd pair of chromosomes, that is in human, is XY, right? Here the X chromosome came from the mother side, the Y chromosome came from the father side, right? Now, because in humans and in human sex differentiating system, male is heterogametic. So in gametogenesis process, when these two chromosomes will separate and they will segregate into two different cells, right? They have to be segregation process in meiosis, right? Because meiosis is a reduction division, and more specifically meiosis 1. So one gamete will get X chromosome, the other gamete will get and obtain Y chromosome. Now let's say for example there is gene A, right? There is some gene, there's an X gene called X gene A. This gene A is responsible, and it has the code that is important for producing a protein that is very important for the spermatogenesis process, right? Now, if that protein is present, then the spermatogenesis process will continue, and it will produce healthy sperms. And if that protein is not present, right, then of course there will be some stop, or it will produce immature or abnormal sperms. This gene is present on, let's say, X chromosome, right, and its counterpart is not present on Y chromosome. In order to and in order for these both sperms, right, to mature into a healthy sperm, let's say here you can see I've written, for example, these two are spermatocyte. This spermatocyte has X chromosome, this spermatocyte has Y chromosome. Now X chromosome has the gene A that is responsible for producing a protein right? And that protein will be used by the in the process of spermatogenesis to produce healthy sperms. Now, X chromosome has this gene. X chromosome will copy that gene. Transcription will happen. Of course, the protein will be translated, and that protein, with the help of these bridges, with the help of these highways, right, and with the help of these connections that are connecting the different spermatocytes with one another, with the help of these junctions, the protein from one spermatocyte that has the X chromosome will be transported to the other spermatocyte that has the Y chromosome, and that the sperm that has the Y chromosome will utilize that protein in order to in order to complete the process of spermatogenesis, in order to in order to become the mature in order to become a mature sperm, right? And after becoming and after differentiating into mature gamete, these cytoplasmic bridges will be ended, right? So that's the key reason. While in females, right, while in female, this is something that is very important. While in female, because females are homogametic, female only produce X and X chromosomes, right? So female will only produce only one type of gamete that has the X chromosome. So female doesn't need these cytoplasmic bridges because females are homogametic. And the male, because they are heterogametic, so they must need cytoplasmic bridges that are connecting the cytoplasm. So that was the reason why in spermatogenesis cytoplasmic bridges are present. Now, the presence of cytoplasmic bridges means the cytokinesis process. That is the final stage of cell division, right? Cytokinesis is the final stage of cell division that happens after mitosis, right? During this process, the cytoplasm divides and one parent cell splits into two daughter cells, right? For example, this is a big cell. In cytokinesis, what will happen? It will produce two different individual separated cells. So in this spermatogenesis, cytokinesis process is incomplete, right? Because the cytoplasm is still connecting, and it's still connected between the two cells. And theoretically, it's very important to understand this process because, for example, in if I talk about birds, so birds have some different sex differentiating system. This is the differentiating system that is in birds. In birds, the male is homogametic while the female is heterogametic, meaning to say the male will produce only one type of gamete, right, that has the particular chromosome, and the female will produce two type of gametes. So theoretically, it's very important that in females and in females that have this type of sex differentiating system that is present in birds, there must be cytoplasmic bridges that are present in the gametogenesis that is in females in birds, right? Theoretically, it's important, but of course, we must see whether the genes and the proteins that are responsible and that are required for the gametogenesis process, whether this gene is present on both chromosomes or on just one chromosome, right? So that was the reason why do we need these cytoplasmic bridges in the process of spermatogenesis and not in the process of oogenesis, specifically in humans. So see you in the next video.